Hi everyone. Today we'll read the second chapter of biology of class 11, that is biological classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of need to use organisms for our own use, like for food, shelter, and clothing. Aristotle was earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system was used till very recently. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organism, and photosynthetic, that is green algae, and non-photosynthetic, that is fungi organism. Classification of organism into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. A need was also felt for including, besides gross morphology, other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of five kingdoms. In the table 2.1, the five kingdom, that is Monera, Protista, Fungi, Plantae, and Animalia, with its character, that is cell type, cell wall, nuclear membrane, body organization, and mode of nutrition is given. So if we talk about the first kingdom, that is Monera, the cell type is prokaryotic and its cell wall is non-cellulosic, that is made up of polysaccharide and amino acid. The nuclear membrane is absent in them. The body organization is cellular. They have cellular body organization, and mode of nutrition is autotrophic and heterotrophic. If we talk about the protista, the cell type is eukaryotic, the cell wall is present in some, nuclear membrane is present, body organization is cellular, that is they have cellular body organization, and mode of nutrition is autotrophic and heterotrophic. If we talk about fungi, the cell type is eukaryotic, cell wall is present without cellulose, nuclear membrane is present, and they have multicellular or loose tissue type of body organization. Mode of nutrition is heterotrophic. If we talk about the Plantae, the cell type is eukaryotic, cell wall is present and is made up of cellulose, nuclear membrane is present, they have tissue or organ level of body organization, and if you talk about the mode of nutrition, they are autotrophic, that is photosynthetic. If we talk about Animalia, cell type is eukaryotic, cell wall is absent, nuclear membrane is present, they have tissue or organ system level of organization, and mode of nutrition is heterotrophic, that is holozoic or saprophytic mode of nutrition. So that is about the different characters of five kingdom, that is Monera, Protista, Fungi, Plantae, and Animalia. Let us look at this five kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, worms, gymnosperm and the angiosperm under plants. The characteristics that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differs in the other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic groups, that is fungi, and the autotrophic green plants, though they also showed a characteristic differences in their wall composition. The fungi had chitin in their walls, while the green plants had cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, that is, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, that is earlier placed in algae within plants, and both having cell walls, with the paramecium and amoeba, which were earlier placed in the animal kingdom, which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdom. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is, based on the evolutionary relationship. In this chapter, we will study the characteristics of kingdoms, Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred as plants and animal kingdoms, respectively, will be dealt with separately in the chapter 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occurs almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot spring, desert, snow and deep ocean where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, pluralis cocci, the rod-shaped bacillus, pluralis bacilli, the comma-shaped vibrium, pluralis vibrio, and the spirillum, pluralis spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from the inorganic substances. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they do not synthesize their own food but depends on the other organism or on dead organic matter for food. First one is Archaebacteria. These bacteria are special since they live in some of the most harsh habitat such as extreme salty areas, that is halophiles, hot spring, that is thermoacidophiles, and marshy area, that is methanogens. Archaebacteria differs from the other bacteria in having a different cell wall structure, and this feature is responsible for their survival in the extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffalo, and they are responsible for the production of methane, that is biogas, from the dung of these animals. Next one is eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. The colonial are generally surrounded by gelatinous sheath. They often form blooms in a polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are the important decomposer. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotic, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human being, crops, farm animal and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by sort of sexual reproduction by adapting a primitive type of DNA transfer from one bacterium to other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Next kingdom, that is kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellate, euglenoids, slime moulds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, the protist cell body contains a well-defined nucleus and the other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. First one is chrysophytes. This group includes diatom and golden algae, that is desmids. They are found in freshwater as well as in marine environment. They are microscopic and float passively in water currents like planktons. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the ocean. Next one, dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella, one lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example gynolax, undergo such rapid multiplication that they make the sea appear red, which is known as red tide. Toxins released by such large number may even kill the other marine animals such as fishes. Next one, euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic, in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on the other smaller organism. Interestingly, the pigments of the euglenoids are identical to those present in the higher plant. Example, euglena. Next one is slime molds. Slime molds are saprophytic protists. The body moves along the decaying twigs and leaves, engulfing the organic material. Under suitable condition, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiates and form fruiting bodies bearing spores at their tips. The spore possess two walls. They are extremely resistant and survive for many years even under adverse condition. The spores are dispersed by air currents. Next one is protozoans. All protozoans are heterotrophs and live as a predator or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoan. First one, amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, that is false feet, as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Second type is flagellated protozoans. The members of this group are either free living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness. Example, trypanosoma. Third type is ciliated protozoan. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity known as gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes a water laden with the food to be steered into the gullet. Example, paramecium. Fourth type is sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, that is malarial parasites, which causes malaria, which has a staggering effects on the human population. Next kingdom is kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. When your bread develops a mold or your orange rots, it is because of fungi. The common mushroom you eat and the toadstool are also fungi. White spots seen on the mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi causes diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotic, example penicillium. Fungi are cosmopolitan and occurs in air, water, soil and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consisting of long, slender, thread-like structure called hyphae. The network of hyphae is known as a mycelium. Some hyphae are continuous tube filled with the multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from the dead substrates and hence are called saprophytes. Those that depend on the living plants and animals are called parasites. They can also live as a symbiont in association with the algae as lichens and with the roots of higher plant as a mycorrhiza. Reproduction in fungi can take place by vegetative means, that is fragmentation, fission and budding. Asexual reproduction is by spore called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores or ascospores and basidiospores. The various spores are produced in distinctive structure called as fruiting body. The sexual cycle involves the following three steps: fusion of protoplasm between two motile or non-motile gametes called plasmogamy, the fusion of two nuclei called karyogamy, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells, that is 2 in number. However, in other fungi like ascomycetes and basidiomycetes, an intervening dikaryotic stage, that is n plus n, that is 2 nuclei per cell occurs. Such a condition is called as dikaryon, and the phase is called dikaryophase of a fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to the formation of haploid spore. The morphology of the mycelium, mode of spore formation, and fruiting body form the basis for the division of the kingdom into various classes. First type is phycomycetes. Members of phycomycetes are found in aquatic habitat and on decaying wood in moist and damp places or as obligate parasites on the plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospore, that is motile, or by aplanospore, which are non-motile. These spores are endogenously produced in sporangium. A zoospore is formed by the fusion of two gametes. These gametes are similar in morphology, that is, they are isogamous, or dissimilar, that is, anisogamous or oogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, and albugo, that is parasitic fungi on mustard. Next type is ascomycetes, commonly known as sac fungi. The ascomycetes are mostly multicellular like penicillium or rarely unicellular, example yeast, that is Saccharomyces. They are saprophytic, decomposer, parasitic or coprophilous, means growing on the dung. Mycelium is branched and septate. The sexual spores are conidia produced exogenously on the special mycelium called conidiophores. Conidia on germination produces mycelium. Sexual spores are called ascospores, which are produced endogenously in a sac-like asci, that is singular ascus. These asci are arranged in different types of fruiting bodies called ascocarp. Some examples are Aspergillus, Claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and buffels are edible and are considered delicacies. Next type is basidiomycetes. Commonly known form of basidiomycetes are mushroom, bracket fungi or puffballs. They grow in soil, on logs and tree stumps and in living plant bodies as a parasites. Example, rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium, plural is basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, that is mushroom, Ustaligo, that is mud, and Puccinia, commonly known as rust fungus. Next type is Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. One perfect, that is sexual stage of the members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by sexual spore known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasitic, while a large numbers of them are decomposers of litter and helps in mineral cycling. Some examples are Alternaria, Collatotrichum and Trichoderma. Next kingdom is kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called as plants. A few members are partially heterotrophic such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant and Cascuta is a parasite. The plant cell have a eukaryotic structure with the prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperm and angiosperm. Life cycle of plants have two distinct phases, the diploid saprophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phase and whether these phases are free living or dependent on the others vary among the different groups of plant. This phenomena is called as alternation of generation. You will study further details of this kingdom in chapter 3. Next kingdom is kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depend on the plant for food. They digest their food in an internal cavity and store food reserves as a glycogen or a fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms shows elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. Next one, viruses, viroids and lichens. In the five kingdom classification of Whittaker, there is no mention of some acellular organisms like viruses and viroids and lichens. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects a virus can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not truly living. If we were understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call virus living or non-living? The name virus, that means venom or poisonous fluid, was given by Pasteur. D.J. Ivanovsky recognized certain microbes as a casual organism of the mosaic disease of tobacco. These were found to be smaller than the bacteria because they pass through the bacterial proof filter. M.W. Bajernick demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and called the fluid as contagium vivum fluidum, means infectious living fluid. W. M. Stanley in 1935 showed that virus could be crystallized and crystal consists largely of protein. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, virus that infects a plant have single-stranded RNA and the virus that infects animal have either single-stranded or a double-stranded RNA or a double-stranded RNA. Bacterial virus or bacteriophage, that is virus that infects a bacteria, are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called as capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric form. Viruses causes diseases like mumps, smallpox, herpes and influenza. AIDS in human is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Diner discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Next one is lichen. Lichen are symbiotic association, that is mutually useful association between the algae and fungi. The algal component is known as a phycobiont and fungal component as a mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provides shelter and absorb mineral nutrients and water for its partner. So close is a association that if one saw a lichen in a nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in the polluted areas. Thank you.